Okay.
Good afternoon. My name is Pavel Abdulkin. I'm the head of Particle Works. Today we'll be talking about encapsulation of hydrophilic drugs, PLGA matrices, or more specifically in PLGA microparticles and nanoparticles. But before we do that, this is the first time we're doing this webinar from within Particle Works laboratories. So I'd just like to take this opportunity to have a quick pan around the laboratories and show you some of the equipment that we're working with here. Yeah. So as you can see, we've got multiple setups running. We've got extraction going on. We've got multiple fume hoods running, and we're running all systems from Dolomite and Cirrus brands in order to create the particles in our particle engineering laboratories. Okay, so what we'll do today is we'll be working with this system to generate PLGA microparticles. We'll do this in three parts. I will go through a presentation on encapsulating PLGA microparticles using Droplet techniques. We will then go through a quick demo of the system, and then I'll answer some questions. So without further ado, let's go to the presentation. Okay, so encapsulating hydrophilic API in PLGA matrices. So before we proceed, let me take this opportunity to introduce the Dolomite particle engineering team. This is a newly formed team within Blacktrace. We are chemists and engineers focused on high-performance nanoparticles and microparticles. We work in biomedical and pharmaceutical industries to create R&D and production equipment. We supply standardized products such as emulsion stabilizers and pre-made nanoparticles, and we also work with our customers to advise them on Droplet microfluidic setups that will solve their issues, and we can also undertake proof of principle studies to verify chemistry working. So why use microfluidics, and what is Droplet microfluidic technology? Well, fundamentally, Droplet microfluidic technology is used to enable reliable formation of highly monodispersed emulsions, double emulsions, Pickering emulsions, multiple emulsions, etc., etc. It can generate a variety of materials at size ranges anywhere between 50 nanometers all the way to hundreds and hundreds of micrometers. This is femto to nanoliter range of droplets in terms of volume. The machinery is highly versatile, and it can be used to create multiple emulsions, encapsulate gas bubbles, create genus particles, as well as multiple other complex structures. And the droplets that are generated using Droplet microfluidic technology can be split, combined, analyzed, diluted, mixed, etc., etc. So it's the fundamental technology that can power a wide range of applications. And as I said up top, we are focusing on pharmaceutical and biomedical applications. So how is microfluidics fundamentally different from techniques commonly used for emulsification today? Well, in a common emulsification technique, you basically have two immiscible liquids, which are sitting on top of each other, and then some sort of a force is applied to mix the two solutions together. Typically, this is propulsion, so some sort of a propeller. It could be agitation via sonication. It could be agitation by some other means, including a whole load of physical means. But fundamentally, you will only ever have statistical control over individual droplets, and you have to put in energy in order to create this emulsion. Now, that's a wasteful and inefficient process, and it can lead to destruction or disruption of API or polymers or proteins that are being put inside the PLGA matrix in this case, or any other droplet. So in Droplet microfluidics, the situation is fundamentally different. We have two immiscible liquids, but they're combined in a controlled fashion at a microfluidic junction, and each droplet is pinched off one at a time in a highly controlled environment. And as the pinching forces are the same each time, each droplet is exactly the same. Now that process occurs at megahertz scale, so it's very, very quick, but you have ultimate control over individual droplets. And as a result, each single droplet that comes off is exactly the same as the other. And also, you are not putting in any force into those droplets, so any of the gentle molecules that cannot be treated with force can be treated in Droplet microfluidics, and that can be dealt with in Droplet microfluidics, and that leads to some unique formulations that we can create in microfluidics and that aren't accessible by conventional methodologies. So another powerful part of Droplet microfluidics is the fact that you can arrange your chips in a variety of ways. So a very basic way of arranging a chip is in a T-junction, and that can lead to creation of slugs, as you can see in the top left image here. Slugs are very useful, particularly if you want them to act as chemical reactors. The emulsified phase inside the slug will never be in contact with the walls of the reactor. So if you have a reaction such as nanoparticle formation, which will lead to precipitation of particles on the sides of the reactor, these are very, very useful. Now, obviously, you can create droplets, and droplets are very useful for formation of particles. So we deal with the second methodology on there quite a lot. In some cases, you want double emulsions or multiple emulsions, and that can be done in droplet microfluidics as well. This could be done using 1 or 2 chip setup, and we will see later on. You can also use microfluidics to encapsulate materials like cells, obviously proteins and API, as we shall see in this presentation. And then also you can use 2 reagent chips to to undertake chemistry. So you've got 2 solutions which are mixed just before the junction. They're pinched off, and the chemistry happens inside individual droplets. So that could be crystallization or some other form of gelation for creation of particles. Then obviously you can make Janus particles as well. We've got two liquids which gel at the interface and then propagate gelation, and so you've got this yin-yang symbol like particle coming off at the droplet microfluidic junction. Now you can also make your junctions at different sizes. So here we've got 3 examples of single junction at the top left corner, and this is a 5 micron all the way to a 50 micron 2 reagent junction. So this would be useful for creation of those 2 reagent chemistry droplets or for creation of the Janus particles, and a micromixer chip, which yes can be used for creation of emulsions at a very high rate, but it can also be used to pre-mix chemicals, and the advantage of using a micromixer chip, as say compared to a batch reactor, is that the mixing times are often order of nanoseconds as compared to seconds in a batch reactor. Now these geometries of chips can be combined to form particles of different nature. So we've got 3 basic emulsions that we'll be looking at today. So you've got a basic emulsion of particles, and these specific ones are alginate beads made in a 100 micron 3D4 chip. These ones are 75 micron. You've got double emulsions, and then you've got multiple emulsions when you've got multiple water droplets on the inside of PLGA beads. Now you can do any of these emulsifications at a range of scales, but we focus on R&D scale, where you're producing grams of material within 1 day at laboratory scale. And typically what you're looking to achieve is you're looking to achieve verification of the process. So you're looking to log down the process, which you will then use at a higher scale. And that scale is achieved using our system called Telos. And in droplet microfluidics, you can scale things up in 3 distinct ways. You can achieve scale up by increasing the flow rates of individual components. You can achieve scale up by increasing the number of junctions working together in the same chip. And you can achieve scale up by increasing the number of chips working together. And the Telos system achieves an order of 100 times scale up from a single junction system by doing all 3 of those. So as you can see in the bottom right-hand corner, the chip has well, in this case, shown 5 junctions, but the chip has 7 junctions working together. The chip, and within the Telos blade, you can have up to 10 chips running together, and the Telos system enables substantially greater flow rates through the chip. So you would typically get of an order of 100x scale up through the Telos system. So how do we make PLGA microparticles and nanoparticles? So obviously this is done in droplet microfluidics, and obviously this is done by controllably combining 2 immiscible liquids at a junction. Now you would control the size of the droplets that you're making by controlling the size of the junction. So if you want to make, say, 30 micron droplets, you'd use a 30 micron junction. If you want to make a 100 micron droplet, you'd use a 100 micron junction. Now within that one junction, you can also control the size of the droplets by varying the relative flow rates of continuous and droplet phase. And as you can see from the 2 pictures here, we've got 1 chip, in this case a 14 micron 3D4 chip, producing 1 micron dichloromethane droplets. Then that same chip can also produce 25 micron dichloromethane droplets simply by adjusting continuous and droplet flow ratios. Now, in a typical particle formation process, the droplets that are formed at the junction would then be cured and set in some sort of way. For example, chemical, thermal, or using UV gelation. Now, in the case of PLGA, things are a little bit more peculiar and perhaps slightly less usual. because what you're doing is you're forming dichloromethane droplets. Dichloromethane contains a certain proportion of PLGA, typically 1 or 2%, but that can go up higher, particularly for low molecular weights of PLGA. So we have previously worked with up to 40% molecular weight PLGA solutions. These droplets are collected and dichloromethane is extracted. And as dichloromethane leaves the droplet, it leaves the PLGA matrix behind, creating a bead. And obviously, the more PLGA you have within the droplet, the higher the size, or the greater the size of the bead is going to be. So in the case of PLGA microparticles, you have an additional tool to control the size of the final beads, and that's the concentration of PLGA. And then obviously, the API is brought in using the droplet phase, and is typically pre-dissolved in the PLGA. So this is spelling out the drying process a little bit more, just to demonstrate an example of this. So we would typically, before starting collection of our samples, we would typically collect a droplet from the droplet microfluidic setup onto a glass slide and observe the glass slide under a microscope. And this is what we typically see. You'd see droplets, which would be quite monodispersed. And as you extracting dichloromethane from these droplets will contract up until they get to their final size, in which case they're still monodispersed, and they're monodispersed beads at this point, at which point we will typically give them a little bit more heat to drive the dichloromethane away. And I think a video spells 1,000 words, so let's see this process in action. This is accelerated 5x time, so this is a process taking place over about 20 minutes. And you're seeing contraction of droplets from 86 microns all the way down to 25 microns, and droplets end up being perfectly monodispersed. Now, it is tremendously important to control this process, as during this process, the API can leak from within the droplet to the outside. And there are various methodologies we employ to do that, and that's typically by controlling the solvents on the outside, that's by controlling the PLGA concentration, and that's by controlling the surfactants that cap the bead. And we can talk about the specifics of individual systems, but it's probably best to address each system individually. So if you have any questions, please contact us directly. Now, in the case of PLGA nanoparticles, the system is fundamentally different. So when you're making microparticles, you're creating an emulsion of two immiscible liquids, typically dichloromethane and water. When you're making nanoparticles, you are using a nucleation and growth mechanism of particle formation. So what happens is your PLGA is dissolved in a water miscible solvent, such as acetone, which is mixed controllably with an aqueous phase containing a surfactant, and that leads to controlled nucleation and growth of particles. We can then control the particle size by adjusting parameters such as concentration of the emulsion stabilizer, residence time, temperature, relative flow rates, etc., etc., etc. There are quite a few levers that one can pull there, but fundamentally, the process is a process of nucleation and growth, where you are nucleating each strand of PLGA polymer individually and letting them self-assemble into a PLGA nanoparticle by controlling the surfactant, controlling the growth conditions. And the API is typically brought in via a acetone line, if this is the case of the hydrophobic API, or it's brought in using the water line, if this is the case of hydrophilic API. But I will talk more about this later on in the presentation. So let's look at three formulations that we have developed specifically for hydrophilic drugs. So why do you need a special formulation for a hydrophilic drug? Well, obviously, humans are made of water. Consequently, most API are soluble in water. And so in order to get the API, water-soluble API inside dichloromethane, tricks are required. So the first trick that we would do is actually to dissolve the API in water and encapsulate that within a PLGA matrix. So the way this is done is we would run two chips. One chip would create a primary emulsion of water inside dichloromethane PLGA, which would then be run again through another chip in a continuous fashion to create a water and oil and water emulsion, as you can see in the image on the right-hand side. And this will trap multiple water droplets on the inside of the PLGA DCM droplet. DCM will then be extracted, leaving the water droplets behind. So the system for this would be typically based on 4 pumps, with aqueous phase coming in to the larger secondary chip, and API and the aqueous phase coming into the smaller primary chip, And then in the primary chip, you're pinching off with PLGA and dichloromethane, and the secondary chip, you're pinching off with aqueous phase. And there's two separate emulsion stabilizers involved, and the chips have opposite coatings to each other. So the 14 micron chip, in this case, is hydrophobically coated. The 100 micron chip is hydrophilically coated, and the entire process is very reproducible and is well controlled using those pressure pumps. So here are our typical results. So top right, top image is an image of emulsion as it comes off, and you can see slight variation in droplet size. This is simply to do with the different states of drying. And when the beads dry fully, you end up with this raspberry-looking like beads, which have multiple water droplets and a little bit of PLGA around this matrix. Now, when you get this inside a human body, PLGA will degrade predictably, and that will release the water droplets from within the bead, and they'll burst outside. And you can see that in the fluorescence microscopy image here, where the API is dyed red and the PLGA is dyed blue. And you can see a layer of PLGA around it. You can see that within the bead, the API is actually quite well distributed. And there's more of it in the center, as you would expect, because perfectly dissolved within the bead. Critically, you're not seeing any API on the outside of the bead. We have measured encapsulation efficiencies anywhere between 50 and 90%, and that's dependent on the API and the PLGA. But obviously, this will vary from one system to another. And that system brings a lot of advantages, such as the ability to pre-dissolve the API before encapsulation and control its concentration, and the ability to control the size of the primary and secondary emulsions. You could go for very small droplets on the inside, and you can go for very large droplets on the outside. It must also be said that in some cases, our customers do want a double emulsion as opposed to a multiple emulsion. So one water droplet on the inside of the PLGA. That can also be done using this methodology. We would simply arrange the emulsion stabilizer for the smaller water droplets to be such that the drying process results in their collapse and agglomeration into one large droplet. And that's by far the best process to create double emulsions, as opposed to trying to do double emulsions within one chip, because frequency matching dictates that production rates will be low, whereas production rates with this system are up to 10 times higher. Okay, so let's look at the second methodology. So the second methodology is direct encapsulation of powdered API within the PLGA bead. Now, this surprises a lot of our customers because they're simply not used to that methodology. And the reason they're not used to this methodology is in a standard batch process, when you have an aqueous system and a organic dichloromethane system, were you to put powdered API into the mixture before emulsification, the API would just go into the aqueous phase, i.e. the continuous phase, and the encapsulation efficiency would be very, very low. However, in droplet microfluidics, the droplets are pinched off individually at the junction, and the entire volume of the line carrying the powdered API is swept, and this gives you the opportunity to trap powdered API inside the PLGA bead. So the setup for this is much simpler. So you simply have one aqueous phase carrier. You've got a droplet phase carrier, which is API suspended within DCM PLGA matrix, and then we typically employ a wash pump to remove any excess material from within the chip at the end of the process and the start of the process. So as I said, this methodology is very, very simple, and the droplet production rates typically are very, very high. So these are the results that we typically get. So these were achieved using a small chip. So these are 2.5 micron PLGA beads. And as you can see from the top image, they're very, very non-dispersed. What you're actually seeing is an SEM image just looking down at a stack of multiple PLGA beads going up. And you're seeing hexagonal close pack, and PLGA beads would not hexagonal close pack unless they're perfectly non-dispersed. And if you zoom in further, then the image in the bottom left is a zoom in on one of the sections of the top image, and you can see all of the beads are perfectly non-dispersed. Now, if you put these same beads into fluorescence microscopy, then the drug that we were encapsulating in this case was doxorubicin, which naturally fluoresces, and you can see that powdered doxorubicin inside the bead, but not on the outside of the bead, and you can see that it's well distributed within the bead. This technique has the advantage of being very, very versatile. It has the advantage of being simple, and has the advantage of having very high encapsulation efficiency. It will typically get between 60 and 95% encapsulation efficiency. But again, this will vary depending on a specific API being used in the powder, the size of the powder being used. So depending on the chip, a variety of powders can be used, but we would typically recommend using powders up to 5 micron for larger chip and up to 1 micron for smaller chips. And obviously, you can pre-mix your APIs, and they will be kept perfectly within the suspension, so you can have multiple APIs and trapped within that same matrix as well. The third methodology is based on the PLGA nanoparticle synthesis. So this is for injectables. Now, to achieve this, we use a 100 micron 3D4 hydrophobic chip. And again, you're going through the nucleation and growth procedure. So you have a polymer solution coming in one line, and you have an aqueous phase coming in the other line. They mix at the 3D4, and you nucleate your particles. Now, that's great, but typically that leads to a problem that PLGA nanoparticles nucleate on the sides of the wall, blocking the reactor. So what we do is we bring in a third component, which is a fluorocarbon oil, which coats the sides of the reactor, much like engine oil inside combustion chamber of an engine, and prevents access of the PLGA nanoparticles to the sides of the reactor. So we typically run half to 2% weight solutions of polymer, and we're able to produce particles anywhere between 50 to 800 nanometers reliably using this 100 micron 3D4 hydrophobic chip technology, and using the 2 emulsion stabilizers and a polymer solution. So the system is somewhat more involved. So you have 4 pumps, obviously, 1 for each phase, and 1 as a wash pump in order to wash any of the materials away from the reactor. And what you find is what we find is we're able to achieve very stable formation of particles that very narrow, with very narrow non-dispersities for time periods exceeding 5 hours. So this is a close to a production technique. Now, previously, we have had our customers reporting back to us that they've been using a micro-mixing methodology of PLGA nanoparticle formation, and that typically leads to results such as the one shown on the top right, where you can make non-dispersed particles, but you also end up with a long tail of particles which are larger than that, all the way up to 1 micron as a result of unwanted agglomeration events. And what we find is that use of this methodology completely cleans up the synthesis, and you only end up with the particles that you want, with none of that long tail present. And this is controllable between, as I said, 50 and 800 nanometers, and work on extending that range further. So some of you have previously asked whether we can achieve 10 or 40 nanometer PLGA particles. The answer is yes, that's possible, but it's worth looking at the specifics of the system, as the process tends to be slightly more involved, whereas this process is very well worked out and works reliably in the lab. So all of the formulations that we've talked about today are enabled by emulsion stabilizers. It's very important to have a reliable emulsion stabilizer in your system. You can look at a variety of academic papers, which would recommend simple emulsion stabilizers such as PVA for PLGA microparticle formation. And that works very well, and that can be easily used inside Dolomite microfluidic systems. However, what we find is if you use more involved emulsion stabilizers, you get substantially improved performance, up to five times of the formation rate. You get substantially enhanced reliability, and critically, you have a system that's guaranteed to work. And when you're talking back to us, we can share results and share the same data. So we've developed 2 emulsion stabilizers currently available on the Dolomite Micro website called Aquaphase, which is a continuous phase emulsion stabilizer, water-based, specifically designed for work with PLGA nanoparticles and microparticles. And Fluorophase is the second emulsion stabilizer that we talked about previously, which is used to stabilize formation of PLGA nanoparticles inside the 100 micron 3D4 hydrophobic chip. These reagents are biocompatible, they're inexpensive, they're cost efficient, and they can be used for scaling up, and they can be used inside pharmaceutical and biomedical processes. So in conclusion, we have multiple methods for fabrication of PLGA microparticles and nanoparticles at a variety of scales, anywhere between 50 nanometers all the way up to 30 microns. We have demonstrated 3 methodologies for encapsulation of API inside PLGA hydrophilic API inside PLGA microparticles and nanoparticles. And all of these methodologies can be scaled up to the Telos system without additional man effort in the scale-up methodology, because you're scaling out as opposed to scaling up. So these methodologies are production ready, and the main advantage of these methodologies are low energy input, high efficiency of API encapsulation, and fabrication of highly monodispersed PLGA beads and nanoparticles. So thank you for your attention. We will go into written Q&A a little bit later, but before that, let's have a look at the system, and let's have a look at the typical result that we can achieve. So we will be looking at formulation number 2, which is direct powder encapsulation within PLGA beads. I'm just put myself tethered here. So we're using this test setup here in Dolomite laboratories. So the entire setup is computer. I'll wait for the camera to readjust. The entire setup is computer controlled. So pressures and flow rates can be controlled individually per each pump. The setup has a spherical plate attached to it, and this is used to keep the API powder in dichloromether suspension, which is then taken through a flow sensor, which we can set to produce droplets at a certain flow rate for each individual pump, which are then flown into a droplet microfluidic chip. In this case, we're using a 50 micron pure reagent chip out there. And the advantage of this compared to a single channel chip is we can bring in additional reagents to the second channel. We can also play around with dilutions, adjust the channel, and don't have to do the second channel if we don't want to. And as you can see on the monitor, if I can transfer the monitor, we're producing highly monodispersed beads, which we can then dry out in the PLGA beads. And of course, no scientific system is complete without a gold standard. Now, you may have noticed that false P pump there. There's a variety of reasons why you want to bring that into your system. For example, in this case, we're actually using this as a reciprocal reservoir, so we're collecting directly into organ or nitrogen. If you have access to that. Alternatively, you might want to collect the droplets into excessive pressure, such as 1 or 2 bar. And it's also very useful if you identify store solutions overnight, which will prevent any evaporation taking place. So I think that concludes the majority of the session. Let's go to the Q&A session.